All right. Hello, everyone. My name is Hamadou. I'm from LMD Lab. I'm a PhD student at Sorbonne University. So today I would like to talk about the synergy between Sun-Induced fluorescence, surface spectral reflectance, and reflectance-based indexes on confined gross productivity. But before we start, allow me to say thank you to my team that I'm working with, that include Yves Goulas, Kamel Soudani, Gabriel Hamimina, and Gwanda Latus. Not even forgetting the young Sans Eco group that allow me to do my presentations. All right, on the frequent changing climate, such as frequent wildfires and heat wave, understanding the role of terrestrial ecosystem in terms of carbon, water, and energy exchanges is important to fill in the knowledge gap on climatic interaction between biosphere and the atmosphere. The terrestrial ecosystem are the main component of the carbon cycle and are highly sensitive to biotic and abiotic stress conditions. Therefore, we urge that an accurate estimation of GPP is real GPP, which is the amount of carbon taken up by vegetation for photosynthesis for a given area and time, is important to quantify the biogeochemical cycles and understand the ecosystem functioning. A GPP can be estimated based on eddy covariance approach locally, but those eddy covariance techniques are limited when it comes to estimate GPP at large scale due mostly to the restricted spatial coverage and distributions. So GPP can also be estimated based on physical and ecophysical modeling methods, but those methods are hampered when it comes to upscale GPP at large scales, mostly due to the lack of understanding of the underlying processes. In recent years, Sun-Induced fluorescence has been used as image as a tool for monitoring GPP at different spatial and temporal scales and across different vegetation types. In contrast to vegetation indexes like NDVI, NIRv, which result from the combination of spectral reflectance and are mostly sensitive to canopy structures and biochemical properties, SIF is a faint signal emitted from the photosynthetic activity. In other words, the light absorbed by the chlorophyll pigments in the range between 400 and 700 nanometers, that's mean on the visible range, are either used for photosynthesis activities or emitted or dissipated as heat, and lastly, emitted in the form of fluorescence in the red and infrared bands, spectrums. Early research based on SIF and GPP relationship, so strong variability depending on the spatial scales and temporal scales, on the vegetation type or biomes, and as well as on environmental conditions. However, we still don't have how SIF is related to GPP across different spatial and temporal scales and across different vegetation types when we go beyond the leaf level. So here, for example, we try to see how to combine SIF measurement, spectral reflectance, and GPP measurement at tower level to see if we can evaluate how SIF is related to GPP and whether SIF and spectral reflectance or vegetation combination would make a better model to predict the dynamic of GPP at the tower level. And lastly, we would like to examine which variable that contribute the most to the GPP predictions. So here the map shows the ICOS flow site location that we use in our studies. And it can be seen that our study site encompasses different vegetation types, including mixed forest, croplands, deciduous broadleaf forest, evergreen broadleaf forest, and so on. We obtained the half-hourly GPP data from the ICOS data portal database. between February 2018 and December 2020. That means of 3 years, mostly of 3 years, full period of data. We are using daily GPP values on the data analysis. So for the remote sensing data that we are also using here is the MODIS Terra and Aqua products and TROPOMI SIF instrument data. For the MODIS provide daily spectral reflectance estimated and corrected for atmospheric gas effect and aerosols. It contains 16 spectral bands, of which the spatial resolution from band 1 and to 7 is 500 meters, and 1 kilometers for the remaining. We are using daily spectral reflectance and vegetation indexes such as NDVI, NIRv, and PRE in our data analysis. The SIF is provide daily estimated near-infrared SIF and this fluorescence, and the spatial resolution is 7 kilometers along track and 3.5 kilometers across track. We use daily ungraded SIF pixel values with center location was less than 5 kilometers from the tower location. For the data analysis, for example, to check the relationship between SIF and GPP at each site and each plant functional type, we feed it a linear regression models. And to test the genericity of the linear relationship, we feed it as well a linear regression model, but on data pool across all sites. So for the modeling part, we established 4 random forest models to predict GPP based on the combination of the available variables. For example, on the first models, we are using surface spectral reflectance values, and on the second, we added to the surface spectral reflectance the Sun-Induced chlorophyll fluorescence, and on the third model here, we are using spectral reflectance, Sun-Induced chlorophyll fluorescence, and a categorical variable to see the effect of the plant functional type on estimating GPP. And eventually, lastly, we used Sun-Induced chlorophyll fluorescence plus vegetation indexes to estimate GPP. On the data, we use 8% of data were used for training and 20% for testing the performance of our model. Some metrics such as the coefficient of determination, the adjusted R squared, and root mean square error and p-values were used to evaluate performance of the models. And we use also the relative importance of each variable to evaluate the part of the contribution of each input to the GPP predictions. Here in this figure, we are showing the relationship between SIF and GPP at each site. For each color code here represent a single plant functional types. What we can see from the figures is the relationship between SIF and GPP was site dependent. And the site dependent SIF GPP relationship can be explained by the heterogeneity of the spatial and temporal variation of the tower footprint, and as well as the difference in canopy structures and the different canopy structures and biochemical properties from site to site, which first can affect the SIF emission, reabsorption, and scattering, and consequently the relationship between SIF and GPP. So here on the left, we are showing the relationship between SIF and GPP based on data pools on the same biome, let's say on the same plant functional type. And what we can see here is the relationship between SIF and GPP was the highest in the DB forest and lowest in tropical forest. And this plant functional type dependent relationship between SIF and GPP is confirmed in this figure, which is on the right, which shows the relationship between SIF and GPP on data pool across all sites. So here we are solving the result of the random forest modeling, or the 4 random forest models, against the observed GPP at the tower level here at each site. And on the right, we have the contribution of each input variable to the prediction of the GPP. But let's focus on the left figures. Here we can see overall, all 4 random forest models could explain GPP with high accuracies. But the strongest model that explained the most GPP with 86% variability of GPP was the model that used only the reflectance. And the lowest were the model that, used the SIF plus the vegetation indexes. And here, from the figure on the left, on the right, we can see that the surface spectral reflectance in the near infrared and red band, as well as SIF and NIRv, are among the most variable that contributes the most to GPP explanation, the GPP estimations. So here we are trying to see if our models, random forest model, can predict, let's say, the seasonal interannual variation of the object GPP. So what we can see from this figure that we can track very well the spatial and the seasonal interannual variation of the object GPP with the different site that we selected here. But something important that I need to mention here is just our models, all our models suffer, for example, to underestimate GPP in some sites and years. For example, here in the wetland, we have all models underestimated maximum of GPP. And here in the grassland as well, in 2020, we have the same situation. So on this result, we are trying to see what example, for example, how our model that include sun-induced chlorophyll fluorescence and reflectance perform at the site level. So here we are comparing the GPP observed and the predicted GPP at each site, right, at each site. So what we can see, we have a high agreement between SIF and GPP overall, even though we have some sites where the prediction is very low, which is mostly related to, we can interpret with the photosynthetic pathway effect, because here in Italy, here in this site, we have, it is a cropland where in our model, we gathered everything, the, let's say, the C3 pathways photosynthetic and the C4 pathway photosynthetic. Here on this, we went on to look at, let's say, at the each plant functional types, what the relationship between SIF and GPP using the same model as well. What we can see here, it's very important and convenient that we see a great agreement between observed and predicted GPP for all the vegetation types that we use on our model. Even in tropical forests, we have higher prediction of GPP. So to conclude, and we saw that the strength of the relationship between GPP and SIF was site and plant functional type dependent. The result also showed that sun-induced chlorophyll fluorescence coupled with reflectance observation explained over 80% of the GPP variability across different ecosystems, but failed to bring new information compared to reflectance alone at coarse spatial scales, not namely at 5 kilometers from the towers. We assume that at these scales, to explore, let's say, the information, the physiological information of information content in SIF is quite a bit hard, because with the spatial scale, seems like that the information of the physical information if SIF is really quite average with, let's say, the structural information content on SIF. So on my ongoing work, we are trying, let's say, to see what the relationship between SIF and GPP at the finite scales. In this regard, we are collecting simultaneous measurement of SIF and GPP at a tower since summer last year. And this year, which is most important, we have the laser-induced chlorophyll fluorescence installed, set up in the same site. So here, our ongoing work is to find out if we can use, let's say, leaf and SIF information to see if we can disentangle the physiological and structural information content on SIF, and as well as to see, because this year is a particular year, where we have a lot of heat wave and even drought effect to find out if we can study the effect of the heat wave and drought on the SIF and GPP relationship, as well as on SIF and leaf relationship. So thank you for your attention.